Hello everyone and welcome. Today we are going to embark on a journey through one of the most vital systems in the human body. The one at very core of nutrition. We will covering the intake of nutrients, especially focusing in the gastrointestinal digestion, absorption, and finally the excretion of waste. This is foundational process that converts the food on our plate into the fuel and building blocks our bodies need to function. So in this lecture we will talk about five sections. The first about the gastrointestinal tract, and then we will talk about the brief overview of digestive and absorptive process, then the small intestine and large intestine, and finally we summarize this chapter. So, introduction. One of the primary considerations for a complete nutrition assessment is to consider the three-step model of ingestion, digestion, and utilization. So on this model, consideration is given to each step to identify all areas of inadequacy or excess. If there is any reason why a step is altered from physical, from biochemical, or behavioral environmental causes, the nutrition providers must select an appropriate nutrition diagnosis for which intervention is required. So intake and assimilation of nutrients should lead to nutritional and overall health. So to understand how to assess nutritional status, we first need a framework. So let's begin with the fundamental three-step models of nutrition. When we want to evaluate person's nutritional health, we look at this simple but powerful three steps model that contain ingestion, digestion, and utilization. So ingestion is simply the act of eating. Digestion is a complex process of breaking that food down, which we'll detail today. And utilization refer to how the cell in our body actually use the nutrient we've absorbed. A problem at any one of these stages can lead to nutritional deficiencies or excesses. So this model provides a logical pathway for us as practitioners, nutritionists or dietitians or physicians to identify where the problem lies and to plan an effective intervention. So the central player in the first two steps, ingestion and digestion, for this model is gastrointestinal tract or GIT. So let's take a look at an overview of this incredible system. The GIT is far more than just simple tube. It's one of the largest and most metabolically demanding organs in our body. So it has the largest and the greatest surface area, where see how in a moment, and it houses the largest concentration of the immune system. This long tube, about 9 meters or 30 feet, stretch from the mouth to the anus and includes all accessory organs like liver, pancreas that are critical for its functions. To visualize how these components fit together, so let's look at the diagram of the digestive system. So here you can see the bath food intake. It starts from the mouth, travels down esophagus to the stomach, yes, then enters the winding tube of the small intestine and large intestine. You can also see the accessories organs, the liver, gallbladder, and pancreas, which produce and secrete substances essential for digestion. Now that we've seen the anatomy, let's talk about the key functions this system performs. The functions of the GIT are diverse. First and foremost, it handles the digestion and absorption of everything we consume, but just as importantly, it acts as critical barriers protecting our internal environment from pathogens and toxins. It's a major hub for the immune system, constantly sampling the environment and coordinating responses. And finally, it's signaling a powerhouse. So communicating constantly with the brain through a pathway known as the gut-brain axis. Gut-brain axis. This is the regulatory signaling to the nervous system, which influences everything from hunger to mood. So to perform its main job of absorption so effectively, the small intestine has a truly remarkable structure. So the inside of the small intestine isn't smooth. It's covered in large folds which themselves are covered in tiny finger-like projection called villi or villi. Each of the cells lining these villi, called enterocytes, has its own set of even smaller projection called microvilli. This intricate layered folding creates an enormous surface area roughly the size of the tennis court, all backed into our abdomen. So this is essential for efficient nutrient absorption. The cell here work incredibly, yes, and not just incredibly hard, and have a very short lifespan from 3 to 5 days, meaning the gut is constantly rebuilding itself. So let's zoom in to get a closer look at this architecture. This diagram shows close-up of the cryptvillus unit. New cells are born in the crypts at the bottom of and migrate up to villi. As they travel, they mature and become fully functional for digestion and absorption. You can also see the capillaries. Yes, look, the capillaries here. You can see, and lymphatic, yes, and lymphatic vessels within each villus, ready to whisk away the absorbed nutrient at to rest of the body, because this lining is so active and renew so quickly. Its health is a paramount and can be easily compromised. The high metabolic rate of the gut makes it very sensitive to our overall nutritional status. Deficiencies, malnutrition, or direct damage can quickly impair its function. A critical concept here is that the gut feed itself. Its cells derive energy directly from the nutrients passing through the lumen. If a person stops eating even for a few days or is fed exclusively through an IV, the intestinal lining can begin to atrophy or shrink. This is the use it or lose it principle of the gut. So use it or lose it. So lose it, so atrophy, exit. Okay, so again, if a person stop eating even for a few days or is fed exclusively through an IV, the intestinal lining can begin to atrophy or shrink. This is the use or lose it principle of the gut, and it's why we prioritize feeding through the gut whenever possible. Now let's put this all together and follow the path of a meal from start to finish. The journey begins in the mouth with a mechanical and minor chemical digestion. In the stomach, food is churned into a semi-liquid slurry called chyme. The real work happens in the small intestine, which is the primary site of digestion and absorption for most of the nutrient. An interesting point is the sheer volume of fluid the gut handles, about 9 liters a day, most of which is from our own secretion. And finally, the large intestine handles the cleanup, absorbing the last bits of water and fermenting fiber. So this diagram that I will show you provide a more detailed map of where these secretions are added and where absorption take place. As you can see, different parts of the tract have a specialized role. Secretion here according to the color, digestion in blue color, secretion in green color, and absorption in pink color. The secretion here are added from the salivary glands, stomach, pancreas, and liver, while some absorption start in the stomach. Like alcohol, the vast majority of all nutrient absorption occurs along the length of the small intestine. So the colon's primary absorptive role is for water and electrolyte. The chemical aspects of this process is driven by a class of protein called enzymes. So digestion is fundamentally a chemical process called enzymatic hydrolysis, where enzymes use water to break down large food molecules into smaller ones. This process is helped along by cofactors like stomach acid and bile. The digestive enzymes themselves come from two main sources. Some are secreted into the GI lumen from organs like pancreas, while the other are fixed in place, embedded in the brush border membrane of the intestinal cells. So let's look at summary of the key enzymes involved. In this table, summarize the major digestive enzyme in the mouth, like here, salivary amylase start in the starch. In the stomach, pepsin tackle protein. The pancreas is real work horse producing a powerful enzymes for all three macronutrient: carbohydrate, protein, and fats. Finally, the enzymes for the small intestine brush border perform the final breakdown steps, getting nutrients ready for absorption. So this complex process doesn't happen out pilot. It is very tightly regulated by both nerves and hormones. So the GIT is regulated by two interconnected nervous systems. The enteric nervous system, often called the second brain. The enteric nervous system is a complex web on neurons within the gut wall that controls most day-to-day functions. The central nervous system, our actual brain, can also influence the gut. This is why stress can cause digestive upset. These nerves coordinate two main types of muscles movement: segmentation, which is mixing motion or movements, and peristalsis, the wave-like contraction that propel food forward. Alongside the nervous system, the GIT uses sophisticated system of chemical messengers or hormones. So it might be surprising to learn that the GIT is the body's largest endocrine or hormone producing organs. Specialized cells called enteroendocrine cells secretes dozens of hormones that act as traffic cops for digestive process. They signal hunger and fullness of the brain, control the rate at which the stomach empties, and even stimulate the growth and health of the intestinal lining itself. So let's meet a few of the most important these hormones. In this table, here there is 5 key players. We will start with gastrin. Gastrin released from the stomach and signal for acid production. When acidic chyme enter the duodenum, secretin is released, telling the pancreas to send bicarbonate to neutralize this acid. So CCK is released in response to fat and protein. It tells the pancreas to release digestive enzymes and the gallbladder to contract and release bile. Mutilin helps with motility between meals, and somatostatin is a general inhibitor acting as a brake on the system. With that background, the key players, let's revisit the digestive journey starting in the mouth and the stomach. So digestion begins in the mouth with the mechanical grinding of food by our teeth and the initial chemical breakdown by enzymes in our saliva. Once swallowed, the food enters the stomach. Here, a powerful muscle contraction mix it with gastric secretion to form a chyme. The K secretion are the hydrochloric acid HCl, which sterilizes the food and denatures protein, and pepsin, the enzymes that start the protein digestion. Here, the pepsin is activated from pepsinogen by hydrochloric acid, begins protein digestion. The stomach also produce intrinsic factor, a substance absolutely essential for absorbing vitamin B12 later on. From a hostile acidic environment of the stomach, the chyme is slowly released into the main event, the small intestine. So the small intestine is where the vast majority of digestion and absorption occurs. As the chyme enters, it's met with a flood of secretion, the pancreas secretion, sent in bicarbonate to neutralize the acid, and cocktail of powerful enzymes to break down carbs, fats, and protein. The liver produces bile. Which is stored in the gallbladder and released to emulsify the fats, breaking large fatty globules into tiny droplets. The final step is carried out by enzymes located on the brush borders of the intestinal cells, which completes the breakdown process. Once these nutrients are in their simplest form, they have to cross the intestinal wall into the body. This accomplished through several transport mechanisms. So there is two basic way nutrients get into our cells. The first is a passive transport, where the nutrients simply move from the area of high concentration in the gut to an area of low concentration in the cell. This doesn't require energy. It can happen via simple diffusion or with the help of the carrier protein, which is called facilitated diffusion. But the second is active transport, and this is require energy. This process requires energy in the form of ATP to pump a nutrient into the cell, often against their concentration gradient. And here the figure clarify the information. Here you can see the cell membrane. Simple diffusion is just the nutrient passing straight through. Facilitated diffusion shows the nutrient being helped across the transport protein. And on the right, the active transport shows the nutrient being bumped across the membrane, a process that explicitly requires energy, requires ATP. Yes, after the small intestine has absorbed most of the nutrient, the remaining indigestible material passes into the final segment on the GIT. There is large intestine. So, the large intestine primary role is to absorb the remaining water and electrolyte, turning the liquid waste from the small intestine into a solid phases. It's also a crucial site for bacterial fermentation of materials we can't digest, like certain fibers. The final product, feces, is mostly water, but the solid component consists largely of bacteria sloughed off large of bacteria, sloughed off intestinal cell, and undigested food. This brings us to a fascinating and critically important topic: the community of microbes that lives, yes, within our large intestine. This is very important for the nutrition. Our gut is home or trillions of microorganisms, bacteria, viruses, and fungi, collectively known as intestinal microbiota or microbiome. They are not just a passive resident. They perform essential function for us. They help protect us from pathogens. Yes. Also stimulate immune function and even produce certain vitamins and beneficial compounds called short chain fatty acid. When this community is out of balance, a state called dysbiosis. It's associated with a wide range of disease. So dysbiosis, imbalance in the microbiota associated with various disease. The composition of this internal ecosystem is shaped by many factors throughout our lives. In this figure, yes, illustrates the microbiomes developed and changes over our lifespan. It is influenced by our genetics and how we are born, our environment, stress level, and especially our diet and use of medication, using drugs like antibiotics. A healthy, diverse microbiome is resilient, but major disruption can shift it towards a state that may promote disease. The good news is that we can actively support a healthy microbiomes through our dietary choice. We can enhance our gut microbiota in several ways. Probiotic are beneficial live bacteria that we can consume in fermented food like yogurt or in supplements. Prebiotic, on the other hand, are types of fibers that we can't digest but that serve as food for our beneficial gut microbiomes or microbes. Good sources include onions, garlic, and whole grains. A product that contains both is called a symbiotic. Now let's take a closer look at the specific digestive and absorptive process for each of the three macronutrients, starting with carbohydrates. Carbohydrates digestion begin with amylase, as I mentioned before, and is primarily carried out by pancreatic amylase in the small intestine. These enzymes break large starch down into smaller sugar. The final breakdown to single sugar unit is a monosaccharide is done by brush border enzyme like lactase and saccharase. Glucose and galactose are then actively transported into the cell via SGLT1 transporter, while fructose enter via facilitated diffusion using GLUT5. All the three then exit into the cell in the blood stream through GLUT2 transporter. And this figure, I think, is easy to understand. Here you can see the large starch molecule sequentially broke down by salivary. Here the pink color is salivary, and pancreatic amylase is green. Yes, into smaller and smaller piece until the final disaccharide here and the disaccharide are cleaved into a single glucose molecules by intestinal maltase right at the cell surface ready for absorption. So next, let's examine how the body digests and absorbs protein. The protein digestion starts in the stomach with the enzyme of the pepsin. The process continues in a small intestine where pancreatic enzymes like trypsin break the large protein chains into smaller fragments called polypeptides. The final breakdown is accomplished by peptidase on the brush border. Interestingly, our bodies don't just absorb single amino acids, we can also absorb small peptides of 2 or 3 amino acids, which are then broken down inside the intestinal cells. Finally, we will look at the most complex of the 3 digestion and absorption of the lipids or fats. Digesting fats or lipids is a challenge because fat and water don't mix. The first step is emulsification, where a bile salt acts like soup to break large fat globules into tiny droplets. This increases the surface area of the pancreatic lipase to work, breaking triglyceride down. These products then get packaged into tiny spheres called micelles, which act as transport shuttles to the intestinal wall. Once inside the cell, the fat are reassembled and packaged into special transport particles called chylomicrons, which enters the lymphatic system, not the blood. The next diagram I will show you and provide a wonderful summary of this entire process. Here in this figure summarize the emulsification digestion of micelles formation and chylomicrons formation process for lipid. Here you can see the whole sequences in action. The large lipid droplet is emulsified by a bile salt. Pancreatic lipase digest the fats, which then form micelles. The micelles deliver the fat to enterocytes, and where they are repackaged into chylomicrons, which then enter the lacteal or lymphatic vessels within the villus. To bring together, let's look at the final comprehensive map of the GIT absorption throughout the GIT. In this figure, there is a full summary. There is a comprehensive diagram showing where all major nutrients, vitamins, minerals, and water are absorbed throughout the GIT. This summary diagram is a great way to review. It shows us that while the stomach absorbs few things, the duodenum and jejunum of the small intestine are where the majority of micronutrients, vitamins, and minerals are absorbed. The ileum has a specialized job for absorbing vitamin B12 and recycling bile salt. Finally, the colon focuses on absorbing water, electrolyte, and the short chain fatty acids produced by microbiota. So let's conclude with the main take-home message from today's lecture. In summary, The conversion of food into into usable nutrient is an incredibly complex and beautifully coordinating process regulated by nerve hormone and enzymes. The gastrointestinal tract unique structure is perfectly designed for this task. We've also seen that maintaining the health of the gut lining and the balance of its microbes or microbiome is absolutely essential for our overall health. When this system is disrupted, it can lead to a host of nutritional problems that we as practitioners will need to identify and manage. Thank you for your attention and goodbye and see you next lecture.